Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Before I start the experiment, I want to explain about what is meant by protein purification process. Protein purification is the separation of a specific protein from contaminants in a manner that produce a useful end product. Why we must purify proteins? Because to determine their structure and study their biochemical properties. In this experiment, I want to perform the experiment about protein purification using column chromatography method. Column chromatography is one of the most common methods of protein purification that is used to separate and purify biomolecules according to their different in specific properties. Next, I will start the experiment. So, this is the protocol of this experiment. This experiment have 9 number of protocol or procedures. So, now start the simulations. Okay, first procedure is pellet the cells. Spin the EC tube in the microcentrifuge for 10 minutes to separate cells from the growth medium. Okay, put the EC1 centrifuge. Now put the balancing tube. Then check the balance. Make sure balance. Alright. Click close. And close. Set time 10 minutes. Then press start. Alright. Wait until 10 minutes. Alright. Okay, after 10 minutes, remove the EC1 tube. Okay, drop the tubes into the rack. Then open the waste collection jar. Open. Then tile tube over the waste collection jar to dump out the supernatant without disturbing the cell pellet at the bottom. Alright. Okay, next procedure is pellet additional cells. First, set the P1000 to 1000 microliters. Okay, set the volume to 1000 microliters. Save the volume. After that, attach a tip. Okay, attach a tip. After that, open the EC2 tube to increase the amount of red fluorescent protein. So we add more cells from the second tube. Then draw up 1000 microliters of liquid culture from EC2 tube. Next, open the EC1 tube and dispense the culture into the EC1 tube. After that, open the biohazard waste container to throw away the micropipette tips. Eject the tips. Okay. Next, spin the EC1 tube for 10 minutes. So put the EC1 tube in microcentrifuge and the balancing tube. Okay. Balance the tube. Make sure balance. Then set time 10 minutes. Okay, press start. Okay, wait until 10 minutes. Okay, after 10 minutes, remove the EC tube. So drop the tube into the rack. Close. Next, open the waste collection jar. Okay, open. Alright, next, tilt the EC1 tube over the waste collection jar to dump out the supernatant without disturbing the cell pellet at the bottom. Alright, next, procedure number 3 is resuspend the cell pellet in EB. EB, which means elution buffer. Now set the P200 to 150 microliters. Now can set the micropipette volume to 150 microliters. Save the volume. Next, attach a tip. Then after attach a tip, open the EB tube and draw up 150 microliters of elution buffer. Alright, draw up. Then after that, dispense the culture into the EC1 tube. Now open the EC1 tube. Then dispense the culture into the EC1 tube. Then eject the tip into the biohazard waste container to prevent contamination. So, next procedure is slice the cells. First, vortex the EC1 tube for 10 seconds to resuspend the cell pellet. Okay, put back in the rack. Attach a tip to the P200. Okay. Then open the LYB tube, which means lyse buffer tube. Then draw up 150 microliters of lysis buffer. After draw up, dispense the lysis buffer into the EC1 tube. Next, eject the tip into the biohazard waste container. Then centrifuge the tube. Okay, put EC1 tube and balancing tube in the microcentrifuge. Okay. Balance the tube. Check balancing. Alright. Set for 10 minutes. Okay, wait until 10 minutes. Okay, done. Close. And after that, set a timer for 12 hours to incubate EC1 tube overnight at room temperature. Okay, set for 12 hours. Okay, press start. And wait until 12 hours. Okay, done. So we can proceed to procedure 5, which is equilibrate the column. Place the waste collection jar under the valve of the chromatography column. Then open the column to drain away the liquid. Okay. make the waste collection just center. Open the column. Alright, done. Okay, after that, drop the tubes into the rack. Set the P200 micropipette to 200 microliters. Alright, set the volume. Save the volume. After that, attach a tip. Okay, attach a tip. And open the EC1 tube. And draw up 200 microliters of supernatant. Okay, after draw up, open the super tube and dispense the supernatant into the super tube. Open the biohazard waste container and eject the tip into the biohazard waste container to prevent contamination. Okay, done. We can proceed to next procedures. Next procedures is equilibrate the soluble fraction in BP. First attach a new tip. Then open the BB tube. Then draw up 200 microliters of the binding buffer. Okay, dispense the BB into the super tube. Now open the biohazard waste container to throw away the micropipette tips. Then eject the tip into the biohazard waste container. Okay, done. Proceed to procedure number 7 is apply the soluble fraction to the column. Pick up the P1000 micropipettes. Open the tip box. Attach a P1000 tip and draw up 400 microliters of the super solution. Okay, save the volume. Draw up 400 microliters of the super solution. Next, dispense the red super solution into the chromatography column. open the biohazard waste container and eject the tip into the biohazard waste container again. Alright, done with that. Place the waste collection jar back under the valve. Alright. Then open the column and allow the liquid to drain into the waste container. After that, close the valve when the liquid is just above the resin bed. Okay, done. Done with that. So procedure number 8 is wash the column. Set the P1000 micropipette to 1000 microliters and attach a new tip. Save volume. Okay, attach a new tip. Then dispense 1000 microliters of the WB solution into the chromatography column. WB solution means wash buffer. Okay, next open the biohazard waste container and eject the tip into the biohazard waste container. Next, open the column and allow the liquid drain into the waste container. After that, close the valve when the liquid is just above the resin bed. Alright, done. We can move to the last procedures is elute and collect the RFP protein. Okay, first using a new tip, draw up 1000 microliters of the EB solution. Alright, draw up the EB solution. Draw up. After draw up, dispense gently down the side of the column and repeat this step. Okay, draw up the EB solution, then dispense gently down the side of the column. Okay, close the EB solution and put the waste contention jar and open the column. Alright. Next, place the open RFP tube under the valve. Okay, put the open RFP. Okay, open the RFP. Open. Now put under the valve. Alright. Okay, next, open the column to capture the RFP. Okay, open the column. Now, close the valve and RFP tube. Close the valve and RFP tube. Close. Alright. Now place the waste collection jar back. Okay, after that, place the waste collection jar back under the valve. Alright. Then, dispense 1000 microliters of the column equilibrations buffer, which means CEB solution, into the chromatography column. Okay, dispense. Okay. Eject the tip into biohazard waste container. Now, attach the tip and draw up the CEB solution and dispense into the column, into the chromatography column. Alright, into chromatography column. Right. Done. Open the column and allow liquid to drain into the waste container. After that, close the valve when the liquid is just above the resin bed. Lastly, repeat this process 3 times before storing the column away. Okay, eject the tip. Now, repeat the process. Okay, first, attach the tip. Okay. Now, open the CEB solution and draw up. Okay. After draw up, dispense into chromatography column and done. Alright. Done. Open biohazard waste container and eject the tip to prevent contamination. Okay, eject the tip. Right. Repeat again. Okay.